Hi, Dr. Navindra.
Hi, can you hear me?
Yes, loud and clear. So are you sharing the session?
Okay, yes, I will.
No, are you? I'm asking you because I think better get Faven or somebody to inform Dr. Emily also.
I put it in the group.
Did anyone reply? I saw also, but I don't know if somebody replied or not. Please update me if you're sharing.
Last time say yes. Sure, I think I will share the session.
Okay, wait. Let me just.
Who else?
Because nobody replied when you asked if you are sharing the session.
Okay, wait.
So that's the thing. Okay, I'm sorry. I have to go to hall 1 now.
Yeah. Okay. Hi.
Bioplex assay development is built on years of expertise with a rigorous SMARTS development approach that ensures best-in-class performance. Confidence in system performance is easy with the integration of the Bioplex 200 system, software, and consumables. Engage in the immunoassay workflow you're familiar with while getting results from multiple analytes in one well with less sample.
Hi, before we start the session, I just would like to ask whether Dr. Phaedros Azora Jem, Dr. Eric Chong, and Dr. Ahmad Faizal they are already in hall 2 now. Is Dr. Eric Chong here? No response, yeah. How about Dr. Phaedros Azora Jem? How about Dr. Ahmad Faizal? Okay, probably we just wait for a few minutes. Okay, now it's 10:25. I think we will start the session now. Good morning, ladies and gentlemen, colleagues and students. Welcome to the 5th International Conference on Molecular Biology and Biotechnology. I'm Dr. Pang from the University of Nottingham, Malaysia. I will be the chairperson for this morning oral session on medical biotechnology and molecular biology. So first of all, I have few housekeeping announcement to make. So please mute your video and audio so as not to disturb the presenters during presentation. You will be given instruction if you need to turn on your video. So if you leave the room and want to get back in, or you want to move to another room, you have to click on the link in the welcome kit or in your program book. And then the technical team will admit you immediately. So if you have questions to ask the presenter, please type the question in the chat box and we will address them in the Q&A session later on. Okay, so without further ado, I would like to introduce our first speaker today, Prof. Dr. Leong Chee On, who is the CEO of AGTC Genomics, Sendirian Berhad, Malaysia. Just a brief introduction on Prof. Leong. Prof. Leong has been working on drug target discovery and novel molecules development through preclinical and clinical trials for many years, particularly in breast cancer research. He has received numerous national and international awards, including the FMD Fellowship from United States, the Institute of Biomedical Science President's Award from UK, as well as the Top Research Scientist Award, Malaysia. Okay, so he is currently the Chief Executive Officer for ATGC Genomics, as I mentioned previously, but I think previously he was working with IMU. Okay. So his presentation title will be on mining large-scale cancer genome for therapeutic and preventive biomarker discovery. Okay, Prof. Leong.
Yes, thank you. Thank you, Yofan. Let me share my screen.
Yes, but Prof. Leong, just a gentle reminder. So your presentation is 30 minutes. So about 25 minutes, I will ring the first bell. Okay, so exactly at 30 minutes, I will ring the bell again.
Yeah, sure. Yeah.
Thank you, Prof. Leong.
Okay. Thank you. Good morning, everyone. So thank you for having me here this morning. So to talk about some of the work that we have done previously in terms of data mining for cancer genome. Now, I think for the past 15 years, I think there have been a lot of progress in terms of cancer research. So, for example, okay, we know that about 5 to 10% of the cancer cases are actually caused by genetic defects, hereditary, and the remaining 90 to 95% of the cancers are actually arrived from environmental and also lifestyle. So these are the spontaneous cancer cases, basically. So, and also, we also know that while cancer is basically a result of a gene mutation. So when the genes is mutated, then the cells get the advantage of uncontrolled self-growth, and that's why you get the cancer. And finally, we also map out basically what are the hallmarks of the cancer. They are all together multiple hallmarks that have been identified. For example, okay, sustaining proliferative signaling, avoiding immune destruction, tumor promoting inflammation, and so forth. But the good news about it is that, well, with the like decades of basic research, so to identify what is the features and the phenotype and also the signaling of the cancer cells. So we now actually have a lot of intervention that could actually target each of these signaling pathway. So for example, okay, EGFR inhibitors have been quite successful in terms of targeting the proliferative signaling in lung cancer, and also and of course immunotherapy have been phenomenal in terms of in recent years in targeting some of the hard to treat cancers as well. So as a result, basically the survival outcome for cancer have been improving quite dramatically over the past 50 years. So for example, in the 1970s, we are talking about the survival rate across all the cancer is about 25%. 1 in 4 of the patients that is diagnosed with cancer will actually survive. 75% of them will actually end up dying, basically. And today, we are talking about half of the patients in general will survive, and hopefully by 2034, we will be able to achieve at least 75% of the patients will actually survive from cancer. But the improvement of the cancer survival rate is actually not even, not across all the cancer type. So some of the cancer are doing particularly well. So for example, okay, prostate cancer, breast cancer, and also Hodgkin lymphoma have improved quite a lot. You can get at least about 90% survival rate, so which is quite good. However, for some of the cancer, for example, okay, glioblastoma, brain cancer, and also including pancreatic cancers, these are still while the survival rate is still very low and have not improved as much, basically. Now, over the past many years, I think we also saw that there is a dramatic falling in terms of the cost of the DNA sequencing, and this have been done with the revolutionary development of the next generation sequencing. In fact, the first human genome took about 13 years and about $3 billion to actually sequence it. And today, basically, any lab or any countries or any individual researcher should be able to afford to sequence a human genome. So, for example, basically, to sequence a whole human genome sequence, basically, we are talking about now less than $1,000. For example, okay, we can we can actually sequence a whole human genome with about $600. If you times 4 or 5, basically that's equivalent to about 3,000 Malaysian ringgit. Basically, it's even cheaper than a handphone. So I think in the longer run, more and more people will be able to sequence their DNA, and more research lab would be able to leverage the data that's generated from the whole genome sequencing. Now, as a result of the drop in terms of the cost of DNA sequencing, I think especially in cancer genome landscape, now we actually have more than 30 petabytes of data. So there's a lot, and from different omics platform, genomics, proteomics, even transcriptomics, epigenomics, and so forth. And now what the field is currently facing is that, well, in fact, we have more data than what we can actually cater. So sometimes I do ask ourselves is that, well, in the cancer researchers, do we actually need to generate even more data, because there are actually already a lot of data that is out there. And the challenge now is basically how to translate all this data that have been generated with some large scale study into the biological insights and also translation into clinical use. So I think the frontiers in terms of cancer research is going to be focusing on how to improve the precision cancer treatment, and how to reduce the cost and time for drug discovery and development, and how to improve early detection of cancer. These are the 3 major questions which I think the cancer researchers will need to focus in the next few years to actually translate the knowledge and the data that we have generated so far. So the first thing I want to talk about is basically how the data that is being generated can actually improve precision and discover of new cancer treatment based on the biology of the tumors. So in 2003, I think we were the first we were amongst the first groups that actually coined the word basal-like tumors and also luminal-like tumors. So what we did that time was actually just taking about 100 primary tumors from the patients, and then we run the transcriptomic analysis. And then from the transcriptomic analysis, we just do a very simple, simple for in today's term, it was quite complicated 20 years ago. So using the unsupervised clustering, and what we identify is that, well, there are 2 major groups of breast cancer. One is what we call the basal-like breast cancer, which are mainly ER, PR, and HER2 negative. The other type is actually the luminal-like breast cancer, which have basically ER and PR are positive. And what is remarkable from this study is that, well, by looking at the gene profiling, it actually highly correlated with the survival outcome of the patients. So for example, the luminal-like breast cancer patients normally would have a much better relapse-free survival and also specific survival compared to the basal-like breast cancer. So these studies have triggered a lot of interest for people start to focusing on looking into the biology of basal-like breast cancer. And there are many attempts that actually trying to develop the drug treatment to target specifically the basal-like tumors. And well, interestingly is that, well, with the decades of investment and also the interest, there are now quite a number of drugs that is available that could target it quite well. For example, PARP inhibitors and so forth. So I think this is one of the early studies that actually demonstrate how the transcriptomic data can actually use to classify patients based on their biological phenotype. So since then, I think in 2006, the United States launched a very ambitious project called the Cancer Genome Atlas Project, which actually completed, I think, about 15 years. It took about 15 years to actually complete. Now the project is considered completed. In the project, basically, they look at 33 different tumor types, okay, involving up to about 11 to 13,000 patients. So what they do is basically each patient's primary tumors would actually undergone genomic profiling to identify the mutation, the copy number variation, gene expression, DNA methylation, microRNA, and also the proteomic, and of course, a wealth of clinical data as well. So the interesting part about this project is that, well, each patient actually have the data across all the multi-omics platform. So you can actually layer it out differently and then analyze it, well, from each particular tumors, the different genotype and also the phenotype correlation. It is a very, very powerful data set. And from this TCGA study, what we found is that, for example, okay, we normally identify tumors based on anatomical site. For example, if you have a tumor that is developed in the breast, then we call it the breast cancer. And if you have a cancer in the lung, then we call it lung cancer and so forth. Now, for the first time, okay, using this the data generated from the TCGA, what we ask is basically, if we disregard the anatomical site, can we actually identify the tumors based on the genetic well, based on the transcriptional signature, for example, okay. But the good news is that, well, in most of the cases, okay, when we just look at the transcriptomic signature, yes, indeed, we can actually segregate and identify the tumors based on the anatomical side. For example, ovarian cancer have a very distinct, genetic signature compared to the endometrial cancer, compared to kidney, and so forth. But what is interesting to find is that there are also a subset of tumors which we classified it based on the anatomical side, but actually share very similar biological signature. So for example, okay, a subset of the bladder cancer was actually have a very similar gene signature compared to the lung cancer. And also for another subset of bladder cancer have a very similar signature with the head and neck and also the squamous cell lung cancer. So what is shown here, and of course, vice versa, okay, if you look at breast cancer, you can actually divide them into the luminal-like or breast cancer and also the basal-like breast cancer. And what we've shown here is basically is that, well, if we know that some of the tumors share the similar biological signature, the question then is, can we actually use the same drugs to actually treat this cancer, okay, especially of bladder cancer, which is considered a rare cancer, okay, and lung cancer, which have a lot of drugs that could treat it. So the question then becomes, can we use some of the arsenal or some of the drugs that we have already developed for lung cancer to treat bladder cancer? And there have been quite a lot of success in repurposing some of these drugs, which I will cover later on. So another project that end up from here is basically is a project drive, basically by combining the genomic data, and we are also using the whole genome-wide RNA interference interrogation to identify what are the driver genes mutations that actually drive the cancer or the cancer phenotype. And also the pan-cancer study, which looked at the single-cell sequencing to actually identify even much more interesting features in terms of the heterogeneity of the different type of cancer. And the end result of this is basically is that now we have the comprehensive genome profiling technology, which you can actually identify 500 biomarkers in a single assay. So if you compare to the conventional way of genetic testing in the clinics, basically, if you have a lung cancer, for example, okay, you will be first tested for EGFR. Then after that, the results will take 7 days to come back, and then you test for ALK. And if all this tested is good, then finally you will test for KRAS. If KRAS is mutated, then all the EGFR inhibitors is not going to be useful. So on and off, the patients with lung cancer will actually take about a month or 1.5 months to get the genetic profile before the treatment is being recommended. This is the conventional paradigm, so which is still practiced in a lot of the countries. But with the comprehensive genome profiling, so what it can do now is basically, because of all the data that have already available, you can actually do a detection of a large-scale mutation by looking at 500 driver gene mutations, and from there you can actually cut short the treatment time, okay, to about the diagnostic time to about a week, and then you can actually save the cost and also give the treatment to the patients in a very timely basis. This is one of the translation for the many years of research in terms of the omics, the genomic landscape of cancer. And what is even more is that you can actually you can actually take the blood and actually detect the circulating tumor mutations in the bloodstream, and that could totally improve the early detection of cancers, because some of the mutated DNA was found to be circulated in the tumors well before it is actually being detected by some of the technology, like endoscopy or even MRI, for example. So all these have been translated quite well into the clinical practice now, basically. And the challenge now basically is how to bring this to the cost that is actually affordable to the general public in Malaysia. And this is what Prof. Asma talking about. Okay, we need to innovate and to bring all this technology in to actually benefit the mainstream public. So I think this is from the precision medicine point of view. I think another angle, another area where the multi-years of the cancer genomics have actually translated into is actually how it revolutionized and also how it changed for drug development and discovery. So, and a lot of the efforts have been now focusing on repurposing of drugs. So this was cited, in 2019, that the cost of developing a new pharmaceutical drug is actually cost about $2.5 billion now, basically. And not to mention is that it takes many, many years. If you start with 100,000 compounds, you might eventually get one FDA-approved medicine. That is only if you are lucky, basically, and the successful rate is extremely, extremely low. So, and to bring this even more challenging is that, conventionally or traditionally, when the drug that is being developed and enter into the market, they actually have protected by patent for many, many years. If you follow the green line, so this is a typical small molecule drug. So basically, the company can actually benefit quite a lot during the patent while the patent is still active. But once the patent is ended, then the sales will then actually drop dramatically because of the generic market that's coming in. So that has been true for many, many years. But if you look at in recent years, basically, it no longer follow the green color curve. Instead, it actually follow the orange color curve here, whereby the sales actually ramp up very, very quick, but most of the time the sales will drop before even the patents have finished. Why? Because new, better drugs would come in faster. So, and therefore it actually, and the end result of this is that, the company have to spend a lot of money in R&D, and then the sales and the lifespan of the sales becoming shorter. And the end result of this is that you actually have the cancer drugs price that are actually increased more than 100-fold in the past 10 years. And this is going to impact a lot, especially to developing countries like Malaysia. So for example, I always talk about is that when we are developing drugs, we always focus on the toxicity of the drugs, but we always forget about the financial toxicity to the patients and the family. In fact, when we are talking about the cancer drug treatment, one cycle can cost, few 100,000 Malaysian ringgit. So most of the time, cancer patients cannot actually afford it. And if they can afford it, after 1 or 2 years, the family would actually become bankrupt, basically. This is something that for us to actually think about, besides the science and developing the new drugs, I think what is important is also how we can actually innovate and reduce the cost of development so that we can bring the cost of the price of the cancer drugs down to benefit more publics. So one way to do that is basically drug repurposing. So, well, as I mentioned, to develop a new drug from the scratch all the way to the clinic and to the market, it takes about 2.5 billion. If we can repurpose a drug, it is estimated that each drug will cost about 300 million on average to develop it. What is drug repurposing is basically that you take an FDA-approved drug which has been used, which has been tested, phase 1, phase 2, and phase 3 have been tested to be safe to be used, okay? And then you repurpose it for different type of tumor. The example that I mentioned just now is that lung cancer have a lot of drug treatments available. So if we can take a portion or fraction of those available lung cancer treatment and then do a clinical trial on bladder cancer, and eventually if it works, that drugs can then repurpose to use for treating bladder cancer. And that would dramatically, okay, reduce the cost of developing a new drug for bladder cancer. So we are just taking whatever is already available on the shelf, basically. So the challenge then is basically how can we actually rapidly discover new indication for the existing drug to do that, and that is a question that, well, that our group have been working on for many years. And one way we try to do is basically on pancreatic cancer. So why we are interested in pancreatic cancer is that, well, it is one of the rare cancer. Many people probably are not, the public probably are not aware of it. They probably only look at breast cancer, lung cancer, which have high prevalence. But pancreatic cancer, on the other hand, is actually one of the deadliest cancer. In a way, is that, well, the 5-year survival rate is only 5%. Meaning is that, well, if anyone who is diagnosed with pancreatic cancer, okay, if there's 100 cancer patients, 95 of them would actually die of pancreatic cancer in the next 5 years. So even though there have been tremendous effort to look to developing new drug for pancreatic cancer, if you look at the survival curve here, okay, by combining different drugs with EGFR inhibitor, it actually doesn't improve the survival curve very much. And as a result, pancreatic cancer is still using the main drugs that is being used to treat pancreatic cancer is mainly gemcitabine, which is being discovered or used since 1970s, and it have not been changed since then. So what we do then, we actually look at pancreatic cancer. The first thing is that we collaborated with international consortium to look at the gene profiling of the pancreatic cancer. And then what we identify is that there are 4 different subtype of pancreatic cancer. And particularly with the blue line here, the squamous type of pancreatic cancer is particularly deadly. So, and therefore our effort have been focusing on targeting the squamous type of pancreatic cancer. And what we do then is basically we take the gene profiling that is being discovered from the primary tumors. In this case here, the squamous type of pancreatic cancer, and we do a reverse engineering to look at the drug signature. Basically, if we find the differential gene expression between the squamous type of pancreatic cancer versus other subtype, the question then is, can we use this gene signature and then to map it to the gene signature of any of the drugs that have been used in the clinic or approved by the FDA? And we use a technique called the connectivity map, basically to actually map the gene signature of the tumors versus the gene signature that's induced by the drugs or inhibited by the drugs. And what we found here is basically is that, well, we identify a number of groups that is particularly interesting. Okay, for example, the SRC inhibitors, okay, EGFR, IGF, all these inhibitors have shown a very significant effect in terms of the transcriptomic profile to reverse the pancreatic cancer gene signature. And interestingly is that some of these drugs have been already tested in the clinic, and particularly what we are interested in is actually the SRC inhibitors, for example, dasatinib and so forth. And we did an in vitro study basically to look at whether by combining these drugs with the existing drugs, gemcitabine, would that actually generate any synergistic effect. And this histogram here basically shown is that the more blue color you see in the contour, meaning that the more synergistic effect that you will see when you combine with gemcitabine. And what we see here is that we tested altogether 6 different SRC inhibitors. So combined with gemcitabine, and what we see is basically in the squamous-like PDAC, you get a very good synergistic effect when the SRC inhibitors is combined with the gemcitabine. And whereas in other type of tumors, basically is that, well, you don't see that kind of synergism that much. So this is one way that, well, we actually work to repurpose the drugs, and of course this still need to be tested in the clinical trial. And the good thing is that there are some studies which are currently conducted using dasatinib, and we are eagerly waiting for the clinical results to come out very soon. So these are some of the ongoing work, so I won't go through in detail basically. And I think what we will continue to work on, we will actually use the computational methods to actually identify new biomarkers for drug therapeutics. So in fact, in the old days, we always start with the cell culture, to test and then to understand the biology. Now, even in our group, we actually do it reversely. We actually look at the data set first and then formulate the hypothesis, then only go to the lab to do the testing. So I think in summary, basically, I think whenever we talk about, okay, the impact of all this genomic data on the cancer tumors, we always look at the big figure like the survival rate and all this across the cancer. But actually, if you want to look at the impact, I think we need to start to look at very small subgroups, because these are targeted therapy. So probably it's going to be very specific and benefit a particular subgroups, but collectively, okay, if we do this persistently, eventually we will be able to achieve the big outcome in general for cancer, basically. So I would like to, and finally, I would like to acknowledge the group from IMU who did most part of the work that I've presented. Okay, some of them have moved on. So Dr. Felicia is now in associate professor in Sunway, and Dr. Mai is in Shanghai, and the rest of the group. So, and this is my new team, basically just started about early this year in the company that we set up to basically translate all these findings, many decades of findings, into the clinical use, focusing on genomics, basically. And finally, okay, this is my contact. I will be happy to take any questions. Thank you.
Okay, thank you, Prof. Leong, for a very insightful as well as inspiring talk. Okay, I think we have 5 more minutes here. Okay, so I think we have a question chat box. Hi, Prof. I would like to ask, currently there are lots of study regarding anti-cancer properties from medicinal plants or any other related plants. So in your opinion, is these studies really help the Healthcare Institute? If it helps, what should the study focus on?
Okay, that's a good question. So this has been a traditional way of drug discovery. So I think it does help, but then we need to look at it in a more systematic way. So in a way is that there are many labs that are doing the same thing. So I do not know how many people have invested their money and looking for Tongkat Ali or Misai Kuching or whatsoever. So I think moving forward, it really needs to have a consortium and collaboration so that we won't doing work that is redundant. What we wanted to look at really is a more systematic approach. Basically, start thinking of creating a chemical library. You can do the screening as what you do. In fact, if you do it in a high throughput manner, it will be much more cost effective. Even in our lab, basically, we already throw away all the 96 well plates. So the one that we commonly use is 3A4 wells. If not, then it's 1536 wells that we are actually using the robotic, basically. So yes, it will still be useful, basically, but Malaysia, particularly for Malaysia, we have a very rich biodiversity, but we really need to come up with a more systematic way to actually curate and also create a chemical library. That is what I would say. So you can easily get, hundreds of millions of compounds out there for you to screen, not only for cancer, but for other diseases as well. And one thing that we can do nowadays, basically, is that when we take a particular compound and then put it into the anti-cancer, test it on cancer. But the question then is, okay, what happened if it is negative? So then you probably shelve it. Actually, you don't have to. So our modus operandi now is basically for each drug, we will put it into one model cell lines, whether it kills, it doesn't kill, then we will actually run the transcriptomic for it. Because once you get the transcriptomic out, then you will actually know what is the effect of this particular compounds or the drugs. And then from there, you can probably predict and know whether this particular compound might be benefit, okay, for anti-diabetic or cardiovascular. It doesn't have to be anti-cancer all the time. So sometimes if it doesn't kill the cancer cell, might be a good sign because the drug is safe, but then you can use it for other purposes. So if you are serious about it, create a chemical library and run a transcriptomic profile for all of this library, then each compound would then be directed or purposed to different disease model. Yeah.
Thank you, Prof. Leong, for a very insightful answer. So we have another question from Larson. So Larson asked, how fast or slow can we achieve personalized medicine using genome sequencing with the affordable cost?
Yes, we are already in it, basically. Well, I mean, the affordable cost is relative, right? Depends on what you meant by affordable cost. So as I mentioned, basically anyone can afford to do a whole human genome sequencing now. Basically, it's just a matter that you want to do it or not. So for cancer patients, for example, okay, it used to cost about 20,000 to actually do a comprehensive, to do all the genetic tests before it reached to the to the targeted therapy. Okay, but nowadays, basically with the comprehensive gene profiling, as I mentioned, you just take one biopsy, not even one biopsy, basically just a few slices of the paraffin block. Okay, and then if we run the comprehensive gene profiling module, so it costs around 5,000 ringgit, basically, and then you can then get all the recommendation for what drugs can be used to treat that particular patients. So this technology is out there. So I hope that on the clinical side, so on the healthcare side, they can be open to all this new technology. In fact, the costs have dropped down quite dramatically. It is not as expensive as we think nowadays. So we can definitely do it. I think in Malaysia, overseas already doing it, Singapore doing it. I don't see why Malaysia can't do it, basically. Yeah.
Okay, thank you, Prof. Leong. I think due to time constraint, we have to move to the invited speaker. But however, if you still have questions for Prof. Leong, please type in the chat box, yeah? So we'll address it later on. Okay, so now I would like to introduce our second speaker, Dr. Tai Gi-jun, who is a Deputy Director for Research and Innovation from University Science Malaysia. So today, I think he will talk about generation of TCR-like antibodies against latent tuberculosis antigen. So Dr. Tai, just a gentle reminder. So your session is 20 minutes. So I will ring the first bell around 15 minutes, just as a reminder. Okay, then the second bell will be exactly at 20 minutes. Okay, thank you, Dr. Tai.
Hi, good morning, everyone. Thank you, Yufeng, for the kind introduction. My name is Dr. Tai Gi-jun. You can call me Gi-jun. I'm basically from the Institute for Research in Molecular Medicine in USM. So the talk that I'll be presenting today is basically on one of our long-term projects, whereby it is the generation of TCR-like antibodies against latent tuberculosis antigens. Now, before we start, let me introduce a little bit on Mycobacterium tuberculosis and TB. So basically, only in 2020, there are 10 million people who are actually ill with tuberculosis, while basically there's 1.5 million deaths in TB as well on the same year. Well, the thing is that we have been talking about COVID-19 and how it's a respiratory disease and how it actually affects the world. We sort of or kind of neglected other respiratory diseases such as MTB and TB. Now, one drastic findings is that basically 24.8% of the world population is basically infected with something called latent TB or latent tuberculosis. Now, before I go a little bit in depth into what latent TB is, let me just run through what is the MTB and the TB cycle. Well, basically you have the tuberculosis here, whereby at the first page, it will, when it infects a human being, you will basically have active infection. Now, if let's say a patient who is diagnosed with active infection, they could either die because of TB, or if they are cured, they will go into a cycle whereby there are still residual MTB, which is within the lungs of the patients. Aside from that, active infection could also directly lead to infection, latent infection, without showing any signs of TB. All right. And what is happening with the latent TB here is basically that the tuberculosis from granulomas, okay, and then they basically stay within the macrophages and also the alveolar cells. Now, this is very crucial for our work because this basically is indicative of what we mentioned earlier on before, whereby 24.8% of the world population has got latent TB. Now, latent TB does not actually show any clinical symptoms, whereby the person is still active, the person is healthy, and there is no representation or any presentation of TB. However, when somebody has got or is being immune suppressed, or if somebody are infected with diseases or have diseases such as diabetes, HIV, cancer, the latent infection basically converts itself into an active infection. And this cycle is basically a very vicious cycle whereby if, let's say, these people have got any of these diseases, it's normally going to lead to death, or it's going to lead to very active infection. Reason being is that the MTB has really built up into the body system, and therefore for it to actually act, it's really it's going to be very harsh. Okay. Now, what we were actually looking about is basically this latent TB here, which is basically within the cell. Now, in order for us to further understand what this TCR-like antibody is, I would first like to explain a little bit on how the antigen presentation and the generation of an immune response actually works. All right. So for a latent TB, okay, the Mycobacterium tuberculosis basically forms granuloma and stays within the alveolar cells or the macrophages. All right. This basically occurs because it's trying to protect itself from the immune system, because as we all know, the immune system will try to get rid of the MTB. Now, when the MTB is staying hidden within the cells, what is going to happen is that it will upregulate something called the 16 kilodalton heat shock protein X. Okay, so this happens because in an environment where it's harsh within the cell, where it's very high in pH and where there is very little nutrient, basically the MTB just goes into a dormant phase. So when it enters into a dormant phase, it will basically stay within the cell. Now, the thing with the cell is that although there is bacteria staying within the cell and it's not, it's still healthy, there is still a possibility of antigens from this TB to be presented by the cell. All right. So what's going to happen is that some of the antigens within this cell will basically be degraded, fused within the portions of our MHC, or the major histocompatibility complex. For human, we call it HLA, the human leukocyte antigen. And it will basically present parts or bits or pieces of the peptides onto the cell surface. Okay, so when this happens, basically the cell, which is the alveolar cell or the macrophages, will be able to show small signs that it's basically infected with latent TB. All right, because all these tiny little peptides that's presented on the cell surface will basically tell the body that there is a certain sort of infection. Okay. So in normal cases, there will not be too much of an immune response against the cell. Basically, reason being is that the peptides or the antigenic peptides that is here is basically normally presented to something called the TCR, the T cell receptor. Okay. Now, in order for us to basically understand the immune response, or where does this TCR-like antibody come from, we will first have to look at the interaction between a T cell receptor with a HLA. HLA here being the major histocompatibility complex of the human. Okay, so an antigen, which is basically a 9 to 10 mer peptide, will be presented by the cell, as what I've shown previously. Okay, and the T cell receptor from a T cell, be it a CD4 T cell or a CD8 T cell, will basically come in contact with the MHC or HLA complex with the antigen. If it's recognized as a foreign antigen, what is going to happen is that a CD8 T cell response or the CD4 T cell response will take place, whereby CD8 will differentiate, CD4 will differentiate, and basically proliferate to cause an immune response. Now, this is actually very different from an antibody-based response, whereby an antibody basically directly interacts with exogenous antigens. For example, here you will be able to see this is a bacteria cell, whereby the antibodies will come and interact with exogenous antigens. Okay, when an exogenous antigen and an antibody interacts, basically the antibody recruits things such as NK cells, complements, and etc. to basically get rid of the bacteria. Now, for our case, whereby it's a TCR-like antibody, this antibody takes place basically interacts with the HLA or the MHC molecule with the antigen. All right. So for our case, what we do is that we try to generate these antibodies so that we are able to detect this particular complex. This complex does not actually cause too much of an antibody-based response, because normally a TCR-like the T cell receptor will basically compete with it. So therefore, TCR-like antibodies does not actually occur in nature that much. All right. So in order for us to generate this TCR-like antibody, which is unnatural, what we use is basically something called the phage display technology. For phage display technology, it is separated into three key phases, whereby phase one, you will have the generation of the phage display library. So a phage display library is basically a phage mate or a virus against a bacteria, where we put in human or any donor B cell epitopes. Okay, so in order to do so, we basically either choose immune donors from human, from animals, or even synthetic synthetic sequences of B cells, and even from healthy donors, whereby we put all their B cell cDNA into the vectors. These vectors will then be transfected into the phage, whereby it could form up to 10 to the 9 different clones, and then these clones will basically have a sort of a library of all the antibodies of the donor. Okay, you can use one donor, you can use multiple donors, you can use donors from animals such as camels, shark, and etc. But in our case, we are basically using a human phage display library. So here at the second phase, you will basically have your your phage display library, whereby then you will pan it, or we call it a biopanning process, against the antigen of interest. Okay, so this will basically take 2 to 3 cycles, whereby we will try to enrich the bound clones, so that we will get a high number of these clones in order for us to then look for for us to go to the next phase, whereby we try to produce the monoclonal antibodies against it. All right. So for our TCR-like antibodies, we use a SDAB, or a single domain antibody phage display library from the Daniel Kreis group. Okay, whereby at first we'll have the HLA with the refolded HSPX antigen. Okay, here, and then we basically biopan it against against the Daniel Kreis library. And after biopanning, we basically enrich and select the phage niche with the binder. Okay, this will basically allow us to choose the gene sequence of the antibody of interest and then clone it into an expression vector, whereby this expression vector will then be transfected or transformed into a expression host, and then we will be able to produce and purify the binder. After the binder has been purified, we basically have a single domain antibody against the target of interest, where here is basically the HLA against with the HSPX antigen. Okay, so based on what we have done, we have actually looked at 3 different HLA, which is HLA-A2, which basically covers 40% of the human population. We have HLA-A11 and also A24, which basically together with A2 covers 70% of the human population in Malaysia or Southeast Asia. So what we got is we got multiple clones. If you notice here, this is just against, hang on, give me a second, the cursor is missing. Okay, where in the first graph here, you will have multiple clones against the 16 kDa A2. Here we have against 16 kDa A11, and here against 16 kDa A24. So all of these clones that were obtained will then analyze with VBase based on sequencing results, and the best clones will basically be selected. So based on the work that we have conducted, we managed to find a single clone against each of the HLA, and the further analysis was conducted in order to select for the clone. So what we found was that we found a particular clone for all of these, and based on the VBase or IMGT database analysis, we found that it's basically of the V gene IGHV3 family, with a D segment of this, a J segment of this, and the 3 main CTRs of this. All right. So if you basically look at all this data, what it is basically it's just telling you the different sequences and how it's basically correlated to the SDAB antibody that we have generated. All right. So this work has actually been completed. For A2, it was completed in 2018. We have it published. And for the A11 and A24, which is crucial for the human, for the Malaysian population, was basically published in 2020. So you'll be asking, sorry, I think I missed out this, whereby we have the Western blot and also the ELISA analysis of the antibodies, whereby you've got the Western blot for binding against the antigen, and also we have the ELISA. So this is for A2, A11, and A24. Okay, so now you most likely be asking, now we have a binder, okay, which is a single domain antibody. What is actually this? This is basically from this part of an antibody. So an antibody is normally Y-shaped, all right? So if you look at our binder, it's basically just a single part like this, whereby that is why we call it as a single domain antibody, all right? So in order for it to be functional, what we went and did was we went and tried to clone it together with an FC sequence, be it an IgG, an IgA, or etc., based on what our application is. And then we fuse it together in order for us to generate a pcDNA 3.1 mammalian expression vector. Okay, so what we have now is basically we have a transient expression system using the pcDNA 3.1 system. We have an intermediary vector which allows for us to clone our SDAB quickly into the different FCs. And we also have stable expression system using the pcDNA FRT, pcDNA5 FRT system. This will basically lead us to this sort of a format of an antibody, whereby we have the binder here, we have a linker, and then most importantly, we have the FC region. Now, the FC region is crucial because you need the FC region for both diagnostics and also therapeutics. So basically, where are we now? For the multiple antibodies that we have for the A2 TB antigen, we've basically generated all of these different antibodies using the transient expression system. We managed to get some purified antibodies from these main 2 antibodies. The rest of it are still in progress. Some of them, we still need to reclone them into different expression system to allow for us to quickly purify them. And based on this, we've done some ELISA, SDSPH, and also the Western blot. So this is based on the full format antibody. Now, one interesting part of the work that we've done is that in order for us to actually detect the antigen, we could basically do an ELISA. But how do we actually detect the presence of the antigen on the cells? So in order to do that, we did something called a cell-based ELISA. So the cell-based ELISA will basically allow for us to detect antigens on the cells. So what we did is that we cultured our target cells in 24-well plates. We incubate them with the peptides. We then put our antibody in. We then, after that, added the IgG-HRP, which is an anti-IgG antibody. And then we do our chromatogenic substrate, whereby we try to develop the color. And this substrate is then, the media is then transferred into a 96-well plate in order for us to obtain the readout. Now, we were successful in sorting this cell-based ELISA out, whereby for the A2 IgG1, we've managed to repeat it 3 times, all with very good results, whereby we were able to get cell-based ELISA reading for the positive control, and also for the negative, we see that it's very clean. So where are we, or what we have actually generated in the past couple of years working on this project? We've basically generated a pipeline, okay, for TCR-like antibody to both diagnostics and also therapeutics, whereby we have the capability of generating the binders, okay, using the phage display library. So not only that we have already generated antibodies against TB, we've also generated multiple novel binders against HPV-16 and HPV-18, which is crucial for the cervical cancer. We also have the transient expression system, which allows us to express the antibodies together with the Fc sequence. Okay, you only need about 2 weeks from transfection to purified protein with a capacity of 30 ml, and you normally get about 1 milligram per ml of antibodies. Okay, so this has been produced. We have basically produced the TB antibody. We also have the stable expression system, whereby the expression system for consistent SDAB Fc can be produced. This requires basically in the region of about 4 to 8 weeks from transfection to selection of clones. And last but not least, we basically generated developed the cell-based ELISA, which is possible for diagnostics. And we are in the midst of generating an ADCC assay in order for us to determine the killing ability of the antibodies based on an in-house NK92 cell line. Okay, so basically our current team consists of a few PhD students. So you've got from left here, which is Ahmad, who's working on an anti-IL-23. Jacqueline here works on the expression systems for the antibodies, both for transient and also for stable. You have Charlie here, who's working on the DCC assay. Matthew is working on a vaccine project. Hua Chang here basically is a PhD student working on the diagnostics and therapeutics using TCR-like antibody. Kelvin, who is also working on who has actually graduated and currently in Northwestern University doing his postdoc, works on the vaccine of the TB. And lastly, Sylvia, who's my PhD student, who is now currently the postdoc here. She was the one who came up or developed the full TCR-like antibody system. So with that, I would like to end the talk. I would like to also acknowledge the organizers of the conference for giving me an opportunity to share with everybody what we have been doing in the lab. Thank you very much. Back to you, Jennifer.
Okay, thank you, Dr. Tai, for the insightful talk. I think we have one question from Dr. Lai. So Dr. Lai was asking, I'm interested to know more about the condition of whole cell infected with latent TB. For example, whole cell mitochondrial activity and changes to the whole cell defense mechanism that should have been able to remove this mycobacterium.
Okay, so I'm not too sure about the whole cell defense system, but what I can say is that normally people who generate or develop latent TB is because of two reasons. Number one is because the immune system of the body basically generates regulatory T cells. So regulatory T cells is basically something that suppresses the immune system. And during the suppression of the immune system, although you see that there is antigen presentation by the MHC peptide, the whole problem is that then you get upregulation of regulatory T cells. This regulatory T cells basically suppresses any T cell based immune response against the APC or the macrophages or the macrophages or the alveolar cells that has the TB granuloma. So that is why it doesn't actually get rid of the MTB infection. So because of that, it is impossible to use basically the cell-based or the cellular-based immune system to get rid of these infected or latent infected cells. So that is why it persists, and that is also why almost 25% of the world population is basically infected with latent TB.
Okay, thank you, Dr. Tai, for the answer. So due to the time constraint, I think we will now move to the oral presentation session.
All right, thank you very much. Thank you.
Okay,
bye-bye.
thank you, Dr. Tai. Okay, so now we will move on to our oral presentation session. Just some housekeeping announcement before we start our session. So all presenters will be given 10 minutes to present. So I will give the first reminder, I'll bell the first ring at the 8 minutes. And the second reminder will be at exactly 10 minutes. Okay, so for the audience, if you have questions for the presenters, so you can type in the chat box. Okay, so without further ado, I would like to invite the first oral presenter, Dr. Lai Hongwei from Kochi University, Japan. So Dr. Lai.
Yes, thank you. I would like to. Is it okay for me to share my screen?
Yes.
Thank you. Can you see it?
Not yet.
Now?
Yeah, we can see.
All right. All right. Okay, let's go. So good morning. I'm Lai, a Malaysian senior scientist currently working in the Center for Photodynamic Medicine, Kochi University, Japan. I graduated from my master's in USM, University of Science Malaysia, and came to Japan in 2016 and took my master's and PhD on photodynamic therapy in cancer at Tokyo Institute of Technology. So today I would like to introduce my research on further improving the specificity of photodynamic therapy known as PDT. And PDT is currently received a lot of attention from the Japanese government and is a highly promising alternative for cancer treatment due to its high specificity with low side effects. So there's no COI involved in this presentation. So cancer is a notorious disease which involves uncontrolled cell proliferation and the ability to spread to other parts of the body due to genetic mutations and lifestyle factors. The study by Malaysia National Cancer Registry report shows that new cancer cases in Malaysia rose by 11.3% within the last decade. So traditional methods of treating cancer are chemotherapies using anti-cancer drugs, radiotherapy using radiation, and surgical removal of cancerous tissue. So although these treatments are well established, they still have some issues such as the drug resistance and specificity. And in order to solve these problems, various alternatives such as immunotherapy, thermotherapy, and of course, for our focus today, photodynamic therapy have been developed. Today, we'd like to focus on photodynamic therapy, PDT. So PDT is a therapy that kills cancer cells following the administration of photosensitizer, in our case, aminolevulinic acids and light irradiation. So an aminolevulinic acid, we usually call it ALA, is a non-toxic essential amino acid naturally found in the human body. It's also an important component in the porphyrin synthesis pathway. ALA can be uptaken by cells and undergo a series of reactions before finally accumulate selectively as protoporphyrin 9, PP9, in the cancer cells. This PP9 will then emit red fluorescence following light excitations and ultimately triggers phototoxicity to the cell. This interesting phenomenon is currently being utilized by scientists and medical doctors in the studies of phototheranostics, which comprises of photodynamic therapy and diagnosis. So, what are the principles of PDT and PDT? PDT and PDT starts off by oral administration of ALA by cancer patients. ALA will then be uptaken selectively and accumulate as PP9 in tumors. PP9 in tumors will then emit red fluorescence, as shown here, following light irradiation. Scientists manipulated these phenomena to be applicable in photodynamic diagnosis, PDD, for fluorescent-guided resections of tumors. On the other hand, light irradiation induces the production of singlet oxygen and reactive oxygen species, leading to cancer cell death. This is the basis of PDT, photodynamic therapy. So since the effectiveness of ALA-PDD and PDT is highly dependent on the concentrations of ALA and PP9, therefore, the understanding of the uptake of ALA is vital in ensuring high effectiveness of PDD and PDT. So, sorry. So, PDD using orally administered ALA has been patented in our center at 2016 and is currently being used to treat patients with bladder cancer in Kochi University. Let us have a look at the video here. This is a tumor being observed under visible light. And and this is the tumor emit red fluorescence when exposed to blue light irradiation. This blue color region shows normal tissue. So the previous tumor was relatively large and therefore quite easily identified. But one moment, please. Yeah, but for this one, it is almost unidentifiable even by experienced surgeons. However, with the addition of ALA, the tumor can be identified with ease. And it is important to note that complete removal of cancer cells is important to minimize the risk of cancer relapse in patients. And here is removal of the cancerous tissue. As mentioned previously, ALA addition leads to the accumulations of PP9-induced phototoxicity against cancer cells. This phenomenon only occurred specifically in cancer cells due to suppressed activity of ferrochelatase in cancer. PP9 will then absorb the photon and became excited, leading to the production of singlet oxygen and exert cytotoxicity onto cancer cells. On the other hand, normal cells are unaffected due to ferrochelatase over here functioning normally. So despite the obvious benefits of PDD and PDT, recent studies have suggested that a certain amount of PP9 was found in normal cells following ALA addition. This may cause undesirable damage to healthy normal cells. Therefore, we move on to study transporters involved in ALA uptake. ALA is being uptaken into the cells through these transporters and undergoes a series of enzymatic reactions and ultimately accumulate as PP9 in cancer cells. So it is known that PEPT1 over here is the main transporters of uptaking ALA in cancer cells, but the roles of other transporters such as PARP1, torrent transporter, GAT2 remain unknown, especially in both normals and cancer cells. So we hypothesize that these transporters might play an important role in uptaking ALA in normal and cancer cells. Moving on to my research. So first and foremost, the expression levels of these transporters, which are involved in ALA uptake, were examined using sets of normal and cancer cells. Here we have lungs, normal and cancer cells, and prostate, normal and cancer cell lines. So we observed that these PEPT1 and PARP1 are highly expressed in normal cells in both cases, respectively, but lowly expressed in their cancerous counterpart. This suggests that these transporters play an important role in ALA uptake in normal cells. Next, using transporters inhibition assay, we hypothesize a significant reduction of ALA uptake in normal cells, but this is quite important. But we would like to reduce ALA uptake in normal cells without affecting their cancerous counterpart. So table 2 shows the changes in PP9 production following the addition of various transporters inhibitors using HPLC. So here we have ibuprofen as PEPT1 inhibitor, tryptophan as PARP1 inhibitor, taurine and GABA as taurine transporters inhibitor and GAT2 inhibitors. So these inhibitors are particularly selected due to their characteristic of exerting low burden to human or being produced naturally in the body. So ibuprofen is a common drug similar to which have similar effects, quite similar effects like paracetamol, Panadol. Tryptophan is naturally found in the human body. Taurine and GABA is naturally found in neurotransmitters in the brain. So we could consider relatively safe. Okay, going back onto the research, coincided with their transporters expression levels, inhibition of PEP-T1 and PARP1 showed significant decrease in PP9 levels in both normal cell lines due to the suppression of airway uptake ability of the transporters. This decrease in PP9 level is not observed in their cancerous counterpart. So they are lowly expressed and therefore the PP9 accumulation is not affected. So these decrease from here, we suggest that the inhibition of highly expressed transporters in normal cells lead to a significant decrease in PP9 production in normal cells, but not in their cancerous counterpart. This would lead to an increase in cancer specificity of ALA-PDT without compromising the PP9 production in cancer cells. I would like to take this opportunity to thank all the individuals stated for the continuous support throughout the research work, and thank you very much for your attention.
Thank you, Dr. Lai, for the insightful talk. Sorry, due to time constraint, I think we have to move to the next speaker. So if you have any question for Dr. Lai, can you please type your question in the chat box? So I would like to invite our second speaker, Mr. Ting Teo Yik from University Kebangsaan, Malaysia.
Thank you for your introduction. Now I'm going to share my screen for the presentation.
Yes, you can. Okay, we can see your screen now.
Good afternoon, everyone. I'm Ting from Embiosis UKM. Today I'm going to present our latest publication on nephrosin. The article can be found using the QR code. And before I introduce what nephrosin is, I would like to briefly introduce what is celiac disease. Celiac disease affects about 1% world population with a genetic predisposition of HLA-DQ2 or HLA-DQN. It's caused by the ingestion of a wheat product such as bread or cereal that contain gluten. Inside gluten, there is one component called gliadin, which is very protein rich, and it's very hard to be digested by human intestine enzyme, and it can bind to the HLA-DQ2 or DQA allele, just the receptor just now, and trigger immune response, and thus cause digestive symptoms such as diarrhea and abdominal pain. Right now, the only treatment or prevention is a strict gluten-free diet. And despite there are claims in market that there are some enzyme supplements that can help with the celiac disease, all of them actually doesn't degrade the gliadin antigen that can cause the immune response. And a recent study found that the proteases from Nepenthes benzoatal feature fluid can actually degrade the antigen that causes the celiac disease. And what is nephrosin? Nephrosin is actually one of the proteases mentioned earlier found in Nepenthes pitcher plant through omic study, and is previously believed to be proteoendopeptidase because of the specific post-proline cleaving activity, which is characteristic of the PEP. What is interesting about this neprosin is that it has optimal pH of 2.5 and optimal temperature, working temperature from 37 to 50, and resistant to most protease inhibitor. Therefore, it could be a very suitable enzyme for enzyme supplement for severe disease treatment. However, very little is known about neprosin other than these biochemical properties, because in natural, the concentration of protease in nephritic picture is very low and very hard to be harvested for crystallography study. Therefore, during pandemic time, we conducted in silico analysis to study neprosin due to lack of access to the wet lab. And first part of the in silico analysis consists of mainly sequence analysis, where we try to extract information from the amino acid sequence of neprosin, such as a signal peptide and a functional domain, and also relationship between neprosin amino acid with other amino acid sequence using BLAST phylogenetic tree, and also multiple sequence alignment. The next part of our in silico analysis is structural analysis, where we use the amino acid sequence without the single peptide for ab initio protein precision, and carry out structural alignment with a non-protein structure on PDB database using DALI. And lastly, we can visualize our result using camera ads. And this is a summary of amino acid sequence of analysis of neprosin. So in short, all the neprosin from Nepenthes ampullaria and AMPRs, and Nepenthes raffaziana and RMPRs, and also our reference neprosin, Nepenthes from Nepenthes pentatera and BMBR, all have a single peptide, color in yellow, highlighted in yellow, neprosin activation domain, color in red, and also neprosin domain, which is color in blue. And inside the Interpro database, there is about 4,200 entry of protein with this domain architecture, which is a neprosin domain, followed by the neprosin domain. And almost all of them are exclusively plant protein. And this is our result from the structural prediction. Out of many methods, we found that there's two methods that produce very similar structure, which is the Rosetta 4 and Alpha 4 2. And when we inspect the result, we found that Alpha 4 2 actually outperformed Rosetta 4 and produced near experimental quality neprosin model, which is suitable for our further analysis, such as protein docking. And these are the example of the neprosin model generated using Alpha 4 2. And we use the two model shown earlier for structural alignment with known protein available on PDB database using DALI server. And interestingly, our neprosin actually matches to glutamic peptidase from family G1, such as acetylglutamate peptidase and asparaglutamate peptidase. And they all have a Z-score above 2, which means neprosin and glutamic peptidase actually have significant similarity. And we use the crystal structure to do structural alignment inside camera S. And these are the alignment. We found that the glutamic peptidase superposed to the neprosin domain of the neprosin. And the neprosin domain actually resemble the active clef of the glutamic peptidase. What is more interesting is that we found that the main catalytic residue of glutamic peptidase as the name suggests, glutamic acid residue, superposed to one glutamic acid residue on neprosin. However, there is no reported specific post-protein credibility of family G1 glutamic peptidase. Therefore, we have to look into other glutamic peptidase family to study further. And we found that the sole member of G3 glutamic peptidase, which is strawberry mottle virus glutamic peptidase unit, actually has reported a post-protein cleaning activity. Therefore, we generated alpha-4-2 model for the SMOB glutamic fatty acids due to lack of the crystal structure and align them. And what we discovered is that the two catalytic residue identified by mutagenetic study of the SMOB study is actually superposed to two glutamic acid residue on neprosin. Therefore, we propose that neprosin is actually a glutamic fatty acid with two glutamic acid residues. And to support our hypothesis, we carry out molecular dynamic study (MD), and we found that the two glutamic residue actually are in low RMSF region, which means they are very stable in the protein structure during the stimulation. And we also carry out a sequence analysis to validate our hypothesis. We found that the two glutamic acid residue is conserved in all neprosin amino acid sequence and is functional according to conserved result. And these two glutamic acid residue actually is highly conserved in representative proteome 35 of neprosin domain. The native neprosin in the picture fluid actually only contain the neprosin domain without the neprosin activation domain. Therefore, autoproteolysis is needed for neprosin to mature and be functional. And much like AGP, low pH conditions such as pH 2.5 is needed for the autoproteolysis to occur. Hence, based on the mass analysis by previous study, we generated mature neprosin structure as shown on the bottom left. And based on the binding pocket analysis using CASP 3.0, I found that only the mature form of neprosin have a binding pocket in the protective active cleft. And our docking result actually agree with the hypothesis because only mature neprosin can bind with the deaminated alpha-1 gliadin, which, as mentioned in first two slides, it can cause the severe disease after binding to the HLA-2 receptor by triggering immune response. And what is even more interesting is that the protein residue of the deaminated alpha-1 gliadin actually bind inside the sub-pocket in the protective active cleft. The sub-pocket is made out of the two glutamic acid residue that we propose is the protective catalytic diet, and also a few hydrophobic amino acid residue, which are often reported to be involved in protein recognition. And this is the proposed neprosin regulatory mechanism in the nephritis picture. First of all, the neprosin protein is transcribed and translated inside the nephritis picture tissue, and it will be secreted into the nephritis picture, and inside the acidic picture fluid, it will autoproteolysis will occur, and the neprosin activation domain will be cleaved off and form mature neprosin. This mature neprosin have accessible protective active cleft with the two catalytic diet for the post-protein cleaving activity. And this is the proposed mechanism of neprosin. We propose that the hydrophobic amino acid residue is involved in recognition and binding of proline via aromatic stacking. And our main catalytic residue, glutamic 169, act as a general acid by donating proton to the carbonyl group of the sessile peptide bond, and also activate a water molecule that then carry out glucophilic attack on the carbonyl carbon atom of the sessile peptide bond. And another water molecule that bind to the GLU system donate a hydroxyl group to the carbonyl group of the sessile peptide bond. And
I believe due to time constraint, I believe you have to stop now.
Okay, sorry.
Yeah.
That's all from me. Thank you.
I think we have some question for you, but I think later on we have a Q&A session, so we can address it later on. I think we have about 15 minutes there. Okay? Yeah, sorry,
Okay, thank you.
Okay, so now I would like to invite our third presenter, Dr. Firdaus Azura Jem from Shiga University of Medical Science, Japan, and Manipur University College, Malaysia, to present a talk. Dr. Firdaus.
Hello, good morning. Thank you.
You can share your screen now.
Okay, I will share my screen now. Okay, is it visible?
Yes, it's visible.
Okay.
Start now.
Thank you, Ms. Yu Fan Huang. I am Dr. Azura from Shiga University of Medical Science and Manipur University College of Malaysia. Actually, this is my PhD work in Japan. So the research is on the cell competition, neuroepithelial cell competition, triggers loss of cellular juvenescence. So it is a question that remains in the development where we can find the apoptosis cells. It is increased in the early development, okay? And it is highly apoptosis occurs during E6.5, and during development, there must be a mechanism that can detect aberrant or defective or mutant cells. Therefore, we puzzle why some cells is being eliminated through apoptosis during development, and what is the mechanism for the elimination of that cells. So we hypothesize that is the cell competition might be the one of the mechanism that contribute to the selection of fitter cells and the elimination of less fit cells. So this is the cell competition. I believe in Malaysia there's not so much study on cell competition compared to in Japan. Okay, the cell competition actually is found or discovered by Gines Murata and Pedro Ripoll 44 years ago, where they found in the Drosophila wing there is one mutant and wild type populate in the Drosophila wing, but during development, the mutant, which is called minutes, is eliminated during development. So what actually cell competition? It is a fitness sensing mechanism which eliminate unfit cells to maintain tissue homeostasis. So I'm illustrate the figure here. The blue color represent the same fitness level. So the cell with same fitness level populate in a tissue, there will be no elimination. And the green cell also has the same fitness level and there will be no elimination in the tissue. But when there are two type of cells with different fitness level, the less fit cell will be eliminated. In this case, the green cell will be eliminated from the tissue. So actually, when there are two cells with different fitness are present in the same tissue, the less fit cells will be eliminated. So in 2016, Ida and colleagues has published and revealed that cell competition has an important physiological significance in our body. It has roles from development to cancer. That is why cell competition is very important. And in 2019, Nature has featured Prof. Yasuyuki Fujita from Hokkaido University. He's one of the scientists that works on cell competition, and his laboratory has published 16 papers on cell competition, including several mutants such as Ras, Scribble, and also they did repeat the D-mix experiment after 26 years of Morata and Ripoll discovery. So the same things happen again by using the D-mix experiment. And the cell competition actually present in various tissue. It has been demonstrated that cell competition present in skin, muscles, gut, and embryo. And in Nature as well, they has published, the Liu et al. Has published that competing stem cells in the aging tail skin of adult mice eliminate stem cells with lower level of collagen, which demonstrate there is cell competition in the skin as well, and this is one of the mechanism for aging the skin. And a novelty of our research, actually, we investigate the role of cell competition in the nervous system, which has not been reported in other studies yet, and we explore the juvenescence of the cells. Here we found that the loss of cellular juvenescence as a mechanism for cells elimination. So what is cellular fitness? Actually, cellular fitness is the ability of a cell to thrive in a given environment. Okay, it is not only an intrinsic property, but determined relative to the fitness of the neighboring or surrounding cells. For example, signaling activity and metabolic rate. Comes to cellular juvenescence, this is quite new. Okay, in 2020, we have performed a transcriptome analysis in young cells of mice from various organs, such as brain, heart, and liver. So from these cells, we identify juvenility-associated genes, which is selectively highly enriched in the young cells. And from the transcriptome analysis, we found out a collection of juvenility-associated genes, later we call as JAKs, which has enrichment function such as in the cell cycle, alternative splicing, acetylation, phosphorylation, ubiquitination, extracellular matrix in the nucleus, nucleoplasm, and cytoskeleton. And these juvenility-associated genes are related to pediatric disease association, for example, progeria, and also overgrowth syndrome or undergrowth syndrome. So these genes has significant role in the pediatric disease associations. So let's go to the results. First, the establishment of the neuroepithelial cell competition model. What we do actually, we have a non-competitive condition and competitive condition, where the non-competitive condition, we use the same cells in for the cell culture, but for competitive condition, we co-culture two different cells. One is blue, which is wild type. One is green, which is RAS expressing cells. So what happened here, we observe the number of cells in the non-competitive condition and competitive condition. So in the competitive condition, the number of green cells here is significantly decreased up to 72 hours. So after DOPS addition means it is expressing the RAS-V12 because we are using the TET-on system. So co-culture of normal, which is blue, with RAS-V12, which is green, activates cell competition because the number of cells are being eliminated is increasing. So the number of RAS-V12 cells is significantly decreased. Second, we observe the competitive in the time-lapse microscope. So this is a competitive condition, two different cells we observe at the time-lapse microscope. And what happened here? The RAS-V12 green one, which is surrounded by wild-type cells, undergoes morphological changes and eventually dies. So I illustrated in the time panel here. So we would like to know what is the type of cell death in the RAS-V12. So we do immunostaining with apoptosis markers such as caspase-3, and after the immunostaining analysis, we quantify the number of RAS-V12 positive with caspase-3. So it is significantly high in the competitive condition, which shows that RAS-V12 in competitive co-culture underwent cell death through non-cell autonomous apoptosis. And then the mechanism underlying the elimination of RAS-V12 cell from competitive condition. So we have explored the cell juvenescence. Then we try to do immunostaining with one of the juvenility associated genes, which is SRSF7. So when we test for juvenility associated gene SRSF7, we found that in competitive condition, it is positively, the SRSF7 is lost in the RAS-V12, which shows that RAS-V12 are losing juvenility. And then the frequency of SRSF7 positive cells in RAS-V12 is significantly decreased. So we can conclude that cell competition trigger the acute loss of SRSF7 in RAS-V12. And what is the mechanism behind the loss of SRSF7? We hypothesize that the loss is through proteasome-mediated degradation. So we try with proteasome inhibitor and MG132, and we found that when we put MG132, the cells are still having SRSF7, as I showed in the arrow. So through the ubiquitination assay, we found that it is really proteasome mediated.
Dr.
Yeah.
Idrus, I think probably you can wrap up.
Okay. So for the effect of SRSF7 to other juvenility associated genes, you also try EZH2, which is one of the juvenility associated genes. And the effect is, so this figure showing that SRSF7 is important for cell juvenility, and it induced the loss of cellular juvenescence. And after the cells undergoing cell death, it is being phagocytosed by other surrounding cells. This is phagocytosis assay. And for the summary, the co-culture of normal and RAS-V12 activate cell competition. Juvenility loss is observed in loser cells, and SRSF7 is degraded via ubiquitin proteasome pathway. And finally, they are eliminated through apoptosis, followed by phagocytosis. I think that's all.
Thank you, Dr. Idrus, for the inspiring talk. So I think if you have a question for Dr. Idrus, I think you can drop your question in the chat box. Yeah, we will discuss later on in the Q&A session. So now I would like to invite our number 4 presenter, Mr. Ng Jun Wei from University Malaysia Sabah.
Hi.
So Dr. Idrus, I think you can unshare your.
Okay. Okay, then.
Okay, thank you. Jun Wei?
Yeah, everything okay?
Yes.
Okay.
I can see your slide. You may start now. Thank you.
Hi, I'm Jun Wei from University Malaysia Sabah. The topic that I will be presenting today is the association of single nucleotide polymorphism with COVID-19 severity in Malaysia. The coronavirus disease 2019 or COVID-19 is an infectious disease caused by SARS-CoV-2. To date, Malaysia has recorded more than 4.5 million of cases and 35,000 of associated deaths. In general, COVID-19 patients demonstrate a great diversity in the clinical manifestations and disease severity, ranging from asymptomatic to severe. Most patients will experience mild to moderate respiratory symptoms, but factors such as older age, male sex, and the presence of pre-existing health conditions could increase the risk for severe COVID-19. However, this risk factor could not fully explain the diverse COVID-19 outcomes, especially the severe cases occurs among young and healthy patients. Therefore, genetic factors such as single nucleotide polymorphism might be responsible for this. So this study aims to determine the potential SNP which are associated with the COVID-19 severity in the Malaysian population using Illumina Global Screening Array and TechMed genotyping assay. So this study was started with the recruitment of 250 recovered COVID-19 patients from Sabah Local Hospital. They were grouped to 3 clinical stages, asymptomatic, mild, and severe, and their health characteristics were recorded. Their peripheral blood were then collected for genomic DNA extractions. After that, the SNP genotyping was carried out using the GSA. For covariate gender, ethnicity, age, and comorbidity were included in the GSA association analysis to determine the SNP related to COVID-19 severity. To further validate the GSA results, the selected SNPs were also genotyped using TechMed genotyping assay in real-time PCR. Finally, the data was analyzed with allelic discrimination plot, Hardy-Weber equilibrium, and risk association analysis using odds ratio to determine the risk for mild and severe disease. So in this study, we found that the mean age and BMI of both mild and severe COVID-19 patients were not significantly different from those of the asymptomatic patients. Moreover, this study also found that about 76% of the patients had at least one morbidity. Of these, the severe COVID-19 patients had the highest percentage of hypertension and obesity. On the other hand, in the GSA association analysis, all the Manhattan plots shows that none of the SNP reached the genome-wide significant level. Therefore, the top 10 SNP with the lowest p-value were determined. Of these, 3 SNP RS923147, 409017, and 1706271 were selected for further validation with TechMed genotyping assay because of their significant association between genders and COVID-19 severity. Furthermore, all the QQ plot shows a deviations of point and a deflations of p-value from the reference line. This can be explained by the great diversity in Sabahan populations and ethnicity. On the other hand, in TechMed genotyping assay, the genotyping of all the selected SNP show 4 distinct clusters of genotype samples in their respective allelic discrimination plots. Moreover, the chi-square test show that all the selected SNP were within the Hardy-Weber equilibrium with their chi-square value below 3.841. In the risk association analysis, the presence of A allele in the RS923147 SNPs provided the patient with a significant protective effect against severe COVID-19. However, the patient with a G allele had a significant higher risk for severe COVID-19. The similar finding was also reported in the gender group analysis for male patients. Interestingly, the protective effect of A allele against severe COVID was also reported in the patients who are aged above 60 and overweight. There have been no previous study performed to evaluate the effect of this SNP on COVID-19 severity. However, we found that this SNP is located within a gene called CD280, which would produce a scaffold protein important for the interactions of immune cells such as T lymphocyte and antigen-presenting cells. So by looking at the role of CD280 in other viral infections such as lymphocytic poliomeningitis, the protective effect of this SNP could be explained by the fact that the CD280 in CD4 T cell could affect the antibody responses towards the infections. The inhibitions of this protein will promote the differentiation of T cell towards the helper lineage, resulting in enhanced control and improved protections from the infections. Moreover, in chikungunya infections, the mutations of CD280 binding motif could attenuate the virus and affect its ability to establish infections. In contrast, a possible explanation for the harmful effect of this SNP was that in other infections such as hepatitis C, the CD280 will be upregulated, which will then stimulate the virus assembly and propagations, as well as other worse disease outcomes. Next, For the RS1706279 SNP, an interesting finding was that the obese patients with a T allele were significantly protected from the mild COVID-19. Similar to the previous SNP, although the effect of this SNP on COVID-19 severity will remain unknown, this SNP is located within a gene called DLGAT2 gene. This gene could produce a membrane-associated protein that may play a role in the nervous system, such as synapse organizations and neuronal cell signaling. So aside from its association with mental disorder, this protein was also associated with non-small cell lung cancer, which was also served as one of the risk factors for severe COVID-19. Next, for the RS409017 SNP, the patient with an A allele had a significant higher risk for severe COVID-19. Moreover, these patients were also more significantly linked with the symptomatic disease. So as there were also no previous study conducted on this SNP, its role in human body as well as in COVID-19 severity will also remain unknown. So in summary, this study found that the severe COVID-19 patient had the highest percentage of hypertension and obesity. Moreover, this is the first study to report the association between this three SNP and COVID-19 severity. For the RS923147 SNP, the presence of an A allele shows a significantly lower risk for severe COVID-19 in all the patients, including those who are above 60, male and overweight. However, the presence of G allele in male patients shows a significantly higher risk for severe COVID-19. For the RS1706279 SNP, the presence of the T allele in obese patients shows a significantly protective effect against mild COVID-19. However, no significant association with COVID-19 severity were reported in the overall analysis as well as age and gender subgroup analysis. For the RS409017 SNP, the patients with an A allele had a significant association with severe COVID-19. However, all the subgroup analysis shows that this SNP was not significantly associated with COVID-19 severity. Therefore, further study with larger sample size are required to clarify and validate the true effect of this SNP on COVID-19 severity, especially in the Malaysian COVID-19 patients. Finally, I would like to take this opportunity to thank my supervisors, Professor Dr. Lee and Dr. Eric Chong, as well as all the patients and medical officers who were involved in this study. Thank you.
Okay, thank you, Junwei.
Yeah, thank you.
Yeah, on time. So I think if you have any question for Junwei, can you please drop your question in the chat box? Yeah. Okay. So now I would like to invite our fifth presenter, Dr. Eric Chong from University Malaysia Sabah, to present his work. Dr. Eric.
All right. Thank you, Dr. Pong. Please allow me to share my screen.
Yes.
I hope you can see my screen now.
Yes.
All right. Okay. Very good morning and salam sejahtera to all of you. So my name is Eric Chong. I come from Biotechnology Research Institute, University Malaysia Sabah. Today I'm going to share with you one of my study entitled An Integrated Study of Obesity Using Metabolite Profiling and Gene Expression. So we all know that obesity is a serious problem globally, with over 650 million of adults are actually obese. So you can see the trend is actually steadily going up now. So when we zoom into Malaysia, we have the highest rate of obesity in Southeast Asia. And the recent MIPS report also stated that 1 in 2 of the Malaysian adults are actually obese. So you can see there are many news that's actually reporting about this. And obesity is associated with many health problems. And the most recently is that we know that when somebody has obesity, it tends to have a more severe COVID-19 symptom. Therefore, understanding the etiology of obesity is undoubtedly crucial. And we think that the study that integrates the metabolite profiling and gene expression analysis in obesity research is definitely needed. So this leads to our objective of this study is to analyze metabolites that are upregulated in the obese subject when compared to normal subject in metabolic profiling. And also we are going to investigate the gene expression profile of the metabolic pathway involved in the obese subject using real-time PCR. So for methodology, so since this study involved the human subjects, so we obtained the ethical approval from MRC UMS, and after that we collect the sample from the subject with written consent, and some of the blood were sent to hematology and also lipid study, and the blood pressure of the subject was measured. For metabolite profiling, so we extracted the plasma metabolites and profile using the LC or BTRAP-MS, and the metabolite data sets were processed and the involving metabolic pathway was analyzed using the metaboanalyze and also the ICMBB software. For gene expression, we isolated the RNA and we do some QC on that. And for the RNA, we also do the reverse transcriptase into cDNA and we perform a standard curve. For the comparative qPCR RNA targeting the genes that regulating the involving metabolic pathway was also performed. So for result and discussion for the blood biochemistry test, these are some components that are significantly higher in the obese subject, including the red blood cell and also white blood cell and triglyceride. For metabolite profiling, after sorting, we obtained 57 features that are upregulating in the obese subject, including the ceramide, phosphoethylolamide, phosphatidylcholine, triglyceride, phosphatidylserine, and also phosphatidylcholine. So these metabolites were subjected to profile analysis, and the pathway analysis using the metabolite showed that the sorted metabolite in the obese subject highly impacted the sphingolipid metabolism, and they are also the main component in the sphingomyelin ceramide metabolic pathway. So we know that the sphingomyelin metabolic pathway is regulated by these two genes, which is the SGMS2 and also the SMPD1. So we are interested to know what actually happened to the expression of this pathway. Therefore, we do the qPCR analysis on this. So before that, we also do some QC of our RNA sample. We can see that they are highly intact with green number more than 6.7. And we also do a standard curve for the Tagman assay. We can see the R square is more than 0.99 and also the efficiency is more than 90%. So they are very reliable to be conducted for the gene expression analysis. So our comparative qPCR analysis showed that the SGMS2 gene was moderately downregulated in the obese group by 0.8254 significantly. As you can see, the green bar here for the obese subject is downregulated when compared to the normal subject. So this actually indicates that the activity of the SGMS2 enzyme in the obese group is inhibited. And this also further explains an abundance of ceramide and also PC metabolite observed in the obese subject in this study. So as we know that the sphingolipid metabolism is very huge, so we only understand part of it actually. So we require, further study are needed to study this metabolism and how it actually affected obesity. So for concluding remarks, this is the first study to suggest that the expression of the SGMS2 gene in the sphingomyelin ceramide pathway is inhibited in the obese subject. That subsequently promoted the accumulation of ceramide and PC. And the data of this study is actually validated in the metabolite profiling also. So we think that the data of this study are useful for future inhibitor discovery in obesity regulation and prevention. These are some of the researchers cited in this presentation. For acknowledgement, I would like to thank the MOHI and also UMS for the grants. And this is the outlook of our institute. We welcome any research collaboration in the future. And we also have a personal research group that you can go into that and look for our current research activity. So with that, I end my presentation here. Thank you.
Thank you, Dr. Eric Chong, for the interesting talk. I think we will leave the question towards the end during the Q&A session. So now I would like to invite our last speaker for the session, Dr. Ahmad Faisal. Dr. Faisal?
Yes, I'm here. Okay, let me share my slide. Can you see my slide?
Yes. Yes, you may start now.
Okay. Okay, good afternoon to everyone. First of all, I would like to thank the organizing committee, the 5th International Conference on Molecular Biology and Biotechnology, ICMBB 2022, for accepting our extract for the oral presentations. So today, basically, I will share some of our research output with regards to the attachment that have been done by one of my non-graduate PhD program from Ahmad Bayero University, Nigeria. So the title for the presentation today is Cytotoxicity Studies of the leaf, stem bark, and root of Bryonadia salisia, Hyper, and JR wood. This is the outline of my presentation today. I will start my presentation with a research background towards acknowledgements. Okay, let me briefly introduce to the research backgrounds of my topics today. So my research is actually focusing on the area of cancer studies. As we know, cancer is one of the major global health challenges with great case on the morbidity and mortality reported worldwide. There is issues with the persistency and toxicity of the therapeutics modalities currently used, which emerged as one of the preeminent public health concerns of the 21st century. Medicinal plants are increasingly gaining popularity as an alternative form of treatment for the cancer nowadays. So with that, we are racing on Bryonadia salisia is one of the such medicinal plants that enjoy the confidence of traditional medicine practitioners for treatment of this kind of cancer, but needed to be evaluated and validated scientifically. So with that, we are actually racing on the plant. So what is the plant all about? As you can see in the figure that I would like to share in the next slide, it's actually an evergreen plant that grows up to 30 meters in height, commonly called as amatome and locally known as Keranja Rafi by Hausa communities of northern Nigeria. It is widely distributed in tropical and subtropical parts of the world, including but not limited to Saudi Arabia, Yemen, Madagascar, Ethiopia, and Nigeria. So with that, the reason why my student who actually undergoing his PhD non-graduate program in UPM, which is he's from Nigeria. And the plant is actually harvested and collected from Nigeria. So the plant is medically used to treat cancer, gastrointestinal diseases, fevers, headaches, arthritis, diabetes, and inflamed wound as well as ulcer, as was reported recently. Okay, this is the image of the plant. It's actually 20 meter high. You can see the image. It's actually located at Kudini, northern Nigeria. So what is the research hypothesis on this plant? So we actually believe that the plant should contain bioactive compound which are responsible for its anti-cancer activities. So with that, the aim of the research is to evaluate cytotoxic effects of the plants on human colon adenoma cell line, which is HT29. Okay, this is the research methodology. So we started the plant collection, which is from Nigeria. We have collected the leaf, stem, bark, and roots. And then we do the plant extraction accordingly, and then fractionations using several solvent, followed by testing on its cytotoxic effect using colon cancer cell line, which is HT29. So what are the tests that has been conducted in order to evaluate its toxic effects? So we have done the first, toxic effect, which is known as MTTAC, followed by microscopy analysis using several staining as well as flow cytometry to check on its apoptosis, stimulation, as well as cell cycle analysis. So with that, I would like to share some of our research output. So according to the extraction that we have done, we have able to extract several parts from, several extract from several parts. So with stem bark scored the highest here, which is 15.36%. Yeah? So based on the extraction that we have performed, we able to actually come up with several extract and fractionation accordingly using several solvents that we have used, yeah, as zinc, acetate, butanol, as well as the aqueous. So this is our results on the MTT assay. So we have been able to show that the extracts as well as the fractions, yeah, able to inhibit the growth of the H2 cell according to the statistical analysis that we have performed using one-way ANOVA done a test. So we able to show that the compound, as you can see, has inhibition ability towards the cancer cell. And then this is the result that we have translated in a graph to show that the trend, when we increase the concentration, we should able to see the toxic effect also increase toward the cell line. Followed by that, we also perform qualitative and quantitative apoptosis assays to look into detail the cytotoxic effect of the extra on the HC29. So what we have found out that the extract as well as the fraction using AOPI staining show apoptosis bodies stimulations. It is actually stimulate apoptosis on the cell line. Most of the extract that we working with actually show an effect on the apoptosis bodies. So you can see. And then we also do quantitative apoptosis. After we perform the qualitative, we actually more interested to further researching on the ability of these extracts toward apoptosis. So we did flow cytometer to look into the stimulation of this extract on the early late as well as necrosis. So we have found that most of the extract actually show an effect on the stimulation of the apoptosis body as well as the necrotic cell. We also has performed cell cycle analysis to see at what paces these extracts show an effects using flow cytometer. So basically, you can see more or less based on the chromatograms. We can see that most of the extracts, particularly the acetate extract, show an effect at sub-G naught paces in terms of cell cycle arrest on HC29. So based on this finding, we believe that, and then we would like to suggest that the extract as well as the fraction that we have processed from the salicylate has certain effect towards human colon adenoma cell line, which is HC29. So with that, these are the references. It's very limited references because this is quite native plant in Nigeria and is not so much research on that. So with that, I would like to acknowledge my non-graduate PhD program who has done this program in UPM, Mr. Liasu. Next to me, on the photo, the front from your left, and then on your right is the student with my research officer, as well at the bottom is the student with one of our co-supervisor. So with that, I end the presentation. Thank you very much.
Thank you, Dr. Ahmad Faisal, for a very good and inspiring talk. So now I will open the session for Q&A. Okay, so I think when I just scroll through the chat box, Okay, we have some question. First of all, the question to Mr. Ting Tick. Okay. The audience would like to ask, how long do you run molecular dynamics simulation? What is the software did you use to run the simulation? Have you run molecular dynamics simulation with ligand binding? Ting?
Hello. So actually, I use a online web server called CAS Flex 2.0 for the molecular dynamics simulation. It took around 1 to 2 days to get result, and we actually haven't tried to use the model with ligand binding for the molecular dynamics simulation yet.
Okay.
I hope that answer your question.
Okay, thank you, Ting. I think you answered the question adequately. So another question from Yasmin also to Ting. Any possible protein crystallization to confirm the putative active site or domain structure?
It's very hard to just collect the harvest the neprosin from, let's say, the picture plant itself for the crystallization because the concentration is very low. Therefore, it's possible if you do recombinant neprosin expression inside like E. coli or yeast, and then you harvest it and carry out crystallography study.
Yeah, I think most of the time people will use recombinant technology, right? If they want to do crystallization, because then you can get in abundance. Okay, as long as the protein is small enough and they don't have any post-translational modification, that will impact the biological effect. Okay, I think we have answered your question. I think Ting have answered your question, Yasmin. Okay, so now we will proceed to the next question from Fei Wen. So Fei Wen, the question is to Dr. Eric. What is the inclusion and exclusion criteria for your study? Do you have a specific time point? For example, before and after meal or any meal restriction plan, okay, for your participants and the meal content before you collect the blood for your metabolites analysis?
All right, thank you, Dr. Pung and also Fei Wen. So I saw many questions towards me actually in the chat box. So I'm sorry because the slide is not so detailed due to time constraint. Actually, before the blood collection, we actually measure the BMI of the subject first to group them into like obese and also the normal. And after that, we ask them to fast overnight before we take the blood actually. So that is the inclusion for our study. For the exclusion is when we also have a consent form that we will access to ask them whether they have some kind of vitamin consumption and also and also like diabetes like that. So that we will exclude from the study. So we only want the healthy obese and also healthy normal need to be included in our study. Yeah. So should I continue to the next question?
Yeah, the next question is about why ceramide and phosphatidylcholine are upregulated in the obese patient. In opinion, why ceramide are upregulated?
All right, this one is actually based on our gene expression study because the SGMS2 in the pathway is actually responsible for the conversion of ceramide and PC into the sphingolipids actually. So based on the study that our SGMS2 is actually inhibited, that is downregulated, that inhibit the conversion of the ceramide and also the PC into the sphingomyelin. That's why it's accumulated in the obese subject. In our study suggests that. Yeah.
So do you think ceramide, over accumulation of ceramide will have some toxic effect, especially on the brain development and some neurological function?
Actually, it's hard to say because I'm not really deep into this one into the neuro side, but I think there is not really there is not really bad impact of that because we can see that obese subject actually can live quite healthily also. Yeah. So I'm not sure about this one, but I think there's not really a bad effect for that. Yeah.
Okay. That's an interesting study. So I know of a very well-known scientist currently located in Brunei. So it's very near your place. He's working on ceramide. Okay, so probably, you can find him later on. I can give you his contact. Okay, so there is another question from Dr. Damani to Dr. Eric. There are studies in 2019 that show inhibition of SGMS2 have protective effect on cardiovascular and glucose metabolism. So in your study, it seems that SGMS2 gene is reduced in obesity instead of increased. So do you have any comments on this observation?
Yes, thank you for the question. So I would say that, obesity involves even the cardiovascular or also diabetes. Actually, they are very complex disease, not only a single gene that can only, control the disease. So in our study actually suggests that the SGMS2 might be, involved in the obesity. But I would say that like I showed just now, the sphingomyelin metabolism is very huge. So we only understand a small part of it. So we hope that later in future that somebody, or maybe we can cooperate with somebody, that we can further in this particular disease, that we can get some more info about this one.
Yeah. Dr. Damani, I hope Dr. Eric has addressed your concern, right? Yes, thank you. Okay, so do you still have any burning question that you would like to ask the presenters before we end the session? Do you still have any burning question? I think in the abstract, right, we have the corresponding email, in the abstract. So if you have any question or if you are shy to ask in this session, you can drop the corresponding authors.
Dr. Pong, there's one more question there.
Huh?
There's one more question for Dr. Phaedrus.
Okay. Dr. Phaedrus, does it mean the competition will lead to eliminating suboptimal cells by the mechanism of loss of cellular luminescence than apoptosis?
Okay, thank you for the question. Sorry, I couldn't finish the presentation due to time constraint. I have lot to tell. Actually, for the cell competition mechanism in the neuroepithelial, we found that the cells, the suboptimal cells or defective cells are being eliminated through apoptosis and then followed by phagocytosis. And then this is through the loss of cellular luminescence. This is correct.
So Feiwen, I hope Dr. Phaedrus has addressed your question. Okay, so I think I have the last question for Prof. Leong. Prof. Leong, are you still here?
Yes, I'm still here.
Prof. Leong, do you think in the future we will have genes on chips in very, very near future?
Genes on what?
On chips. So like on our IC. They will carry, wherever we go.
Yeah, I think so. Well, if you look at Oxford Nanopore, they already have the Minion, which can actually, like the size of a USB port. You basically just need to prepare your sample, plug the chip, onto your USB computer. Yes, then it will actually do the sequencing for you already. Yeah. So of course, currently the capacity is only to the level of microbial. So you will know as all this nanotech is evolving. So when you can load more and more flow cells, into a smaller piece of device, then you might be able to achieve, you can just have a drop of blood and then you can do all your own whole genome sequencing at home.
Do you think it's good or bad, if we know our disease or probably, some of the inherited gene, bad genes, in ourselves. Do you think it's good or bad?
Well, again, I think it's good. So honestly, as a scientist, well, we always want to seek for the truth, right? So obviously, whether you want to know it or know it, the fact is that it is already there, right? So, well, yeah, sometimes we might be fearful to learn the truth, but again, being fearful doesn't change the fact that you have a certain disease, or you might not have certain disease risk disposition preposition, right? So either way, I think it's a two sides of the coins. You might find out something that you have been worrying all this while is actually not true. You actually don't have to worry about. You know, for example, someone who has a high family history of developing breast cancer, you might be worrying about. You might, you know, consider whether to have kids or not. But again, when you tested it, you might not have it, and that will be priceless. So it doesn't have to be always be very grim about, you know, finding out new disease. It could be finding out that you are perfectly normal. Yeah.
Okay, we thank Prof. Leong. Okay, I think now is 12:36. Okay, so I think we are just on time, okay, for the session. So I hope you really like the session that we plan out for you on medical biotechnology and molecular biology. So now I think before we break for lunch, I would like to remind all the presenters, if you are presenting in this coming 2 p.m. oral session, can you please go into the respective breakout room 10 minutes before for the chairperson, you know, to actually take your attendance. Okay, with that, I would like to thank everybody for the participation, and bye-bye. I'll see you again later in 2 p.m. Okay, bye-bye.
Okay, bye-bye.
Bye-bye.
you.